I'm Cindy Morris, a plant pathologist and microbiologist at France's National Research Institute for Agriculture, Food, and Environment. 2020 is the International Year of Plant Health, and so it's an opportunity to highlight efforts to promote plant health. Here in France, we are promoting research and development to find ways to keep plants healthy, all while reducing or even eliminating the use of synthetic pesticides in crop production. 2020 has also been a year of social and environmental movements to make for a world that is more sustainable and just. These movements have arisen from the convergence of the climate crisis, the COVID-19 pandemic, growing poverty, and racial injustice. Amidst all of this, those of us in agriculture, from research to production, need to keep innovating to find ways to reduce the environmental footprint of agriculture, all while assuring food security. Now, how can we promote this innovation? One way is to take a different perspective on the main ideas and concepts that we use to think about plant disease. In the future, to assure that plants are healthy without relying on pesticides, we will be using many concepts from ecology and those that tell us about how microorganisms change as they interact with plants. Many of these concepts have been developed based on observations from agriculture. And that seems like the obvious thing to do. But I think it is blindsiding us, and it is calling for a new way to look at things. So in this talk, I'm going to present a different point of view from a time frame of about a couple of 100 million years before the advent of agriculture. Then I'm going to tell you how I think that this perspective can help us have new ideas about how to manage plant health. Hey, you guys, this is serious. Yes, I know I might make some mistakes, but we need to talk about these questions. So I'm going to try to go on, and I hope that people will judge for themselves if I've made some mistakes. So now where was I? Perhaps some of you have opened this video just to hear the song at the end. So if you really want to just hear the song, you can go off to the last 3 minutes of the song. But I encourage you to first listen to the lecture because it will help you better understand the lyrics of the song. So here we go. To help you follow the ideas I will present, I will start with a quick reminder of the major geological and biological events that have shaped life on Earth. There are hundreds of sources of information about Earth's history. You can start with Wikipedia if you want more details, but I'm not going to list all of the references that I used. Planet Earth was formed about 4,550 million years ago, abbreviated MYA on the slides. A million years is 10 to the power of 6, whereas GY on the slides means gigayears, or 10 to the power of 9. The first life forms were single-celled organisms that we call eubacteria and archaeobacteria, or simply archaea. They appeared very soon after the formation of Earth, about 4,000 million years ago. Some of these organisms developed the capacity to use the energy of light to transform atmospheric CO2 into oxygen. In other words, they developed the capacity for photosynthesis about 3,200 million years ago. The oxygen that they were releasing via photosynthesis didn't start to appreciably accumulate in Earth's atmosphere until about 1,000 million years later, causing the onset of what is called the Great Oxygenation Event. The progressive accumulation of oxygen in the atmosphere set the stage for the emergence of the life forms that we know today that respire oxygen. The first eukaryotes emerged about 1,500 million years ago. Eukaryotes are organisms whose cells contain subunits called organelles that are distinctly delimited in the cytoplasm by membranes. Nuclei, chloroplasts, and mitochondria are examples of organelles. Eukaryotes would go on to evolve into organisms with a vast array of morphological forms, leaves, roots, flowers, arms, legs, fingers, noses, whereas the eubacteria and archaeobacteria have very limited morphological diversity, most probably because they don't have the energy factories to make this possible. From the eukaryotic ancestor, fungi and oomyces emerged about 1,000 million years ago. Land plants and insects about 450 million years ago. Then land vertebrates and mammals, and the non-avian dinosaurs came afterwards, and the species Homo sapiens diverged not more than about 300 million years ago, and maybe just 100 million years ago. Biological evolution on Earth has been accompanied by changes in the abiotic conditions, and in particular temperature and the concentration of oxygen in the atmosphere. Before our present epoch, there were 4 other periods when persistent masses of ice, in other words glaciers, were present on Earth. These are indicated by light blue bars. Outside of these periods, the overall temperature was too hot to allow ice to persist. The concentration of oxygen in the atmosphere increased up to 30% or more by 300 million years ago and then descended to its current concentration of 20%. The pieces of Earth that form the continents have also been in constant movement. Here you see a cartoon animation of how the continents have moved from 450 million years ago, about the time when plants first came onto the land from their aquatic habitats, until present. What do we know about the first land plants? Here's an artist's rendition, based on fossil evidence, of the morphology of the first land plants. I have also included an image of Earth's continents to indicate how they were oriented at the time plants began to colonize land. These plants had no roots. The cell walls had the same sticky polysaccharide called pectin that holds cells together in modern plants. The outer cells of these new land plants were covered with waxy cutin as are modern plants. These plants also produce molecules that are involved in signaling in modern plants, including strigolactones and enzymes such as lysin motive receptor-like kinases and various protein kinases. From those beginnings arose the vascular plants, the tracheophyta, from ferns and mosses to gymnosperms and angiosperms. Some of the well-known orders of angiosperms are listed here in green. This diversity arose as the concentrations of oxygen and carbon dioxide in the atmosphere varied and as asteroids pelted Earth's surface. These past 450 million years of plant evolution were also marked by mass extinctions. 5 mass extinctions occurred over the past 450 million years. The mass extinction at about 66 million years ago was the only one of these that was clearly due to an extreme asteroid impact. This is the well-known Cretaceous Tertiary, or KT, mass extinction event that eliminated about 75% of all species living at the time, including the dinosaurs. When the dinosaurs disappeared, many smaller herbivores proliferated and diversified. This was associated with a rise in diversity of the plants that were grazed by these new animals, and particularly the grasses. Now we are going to look at the timeline of microbial interactions in terms of how organisms from different kingdoms interacted. Kingdoms refer to a taxonomic distinction between major groups of organisms. The interaction of microorganisms with plants during the process of disease is one example of interkingdom interactions. If we look at all of the different types of interkingdom interactions that occur among organisms, what has been the general tendency for their evolution? Over time, has the intimacy between organisms during these interactions increased or decreased? Have organisms gained functions or lost functions to achieve these interactions? The first interkingdom interaction of which we are aware was the fusion of a bacterial cell with that of an archaea 1,500 million years ago to form the ancestor of eukaryotes. This process of fusion is called endosymbiosis. The fusion of bacteria with archaea, referred to here on this slide as the protoeukaryote, led the bacterium to literally settle down into the protoeukaryotic cell and eventually become the chloroplasts of plant cells and the mitochondria of all eukaryotic cells. Mitochondria provided the energy that led to the morphological diversification that was not possible in the prokaryotes. For more details about the exciting processes that led to the emergence of eukaryotes and their morphological diversification, I highly recommend Nick Lane's book, The Vital Question. The next significant interkingdom interaction that developed was the penetration of fungi into the interior of plant cells, in other words, into the lumen. Arbuscular mycorrhizal fungi established intimate relationships with plants as early as 400 million years ago. Hyphae of these fungi, that are also called endomycorrhizae, penetrated into the plant cell lumen where they formed complex structures, arbuscules and vesicles, that are used for nutrient exchange and storage. The plasma membrane of the plant cells surrounded the penetrating hyphal filaments and the arbuscules and vesicles as they formed. How was the age of this interaction estimated? In fact, fossil evidence played an important role. It is rare to have fossil evidence to date the age of microorganisms, but arbuscular mycorrhizal structures were found in the fossilized bits of the primitive land plant Aglaophyton major in sediments in Scotland. These fossils contribute to the confidence that the scientific community has about when the arbuscular mycorrhizal associations emerged. The rapid appearance of terrestrial mycorrhizal interactions soon after plants arrived on land is not surprising, because the stage had been set several hundred million years previously. Terrestrial mycorrhizae arose from pre-existing mycorrhizal interactions in aquatic environments that have been dated to have emerged about 700 million years ago. If we look at other fungi that interact with land plants, there are quite a few that deploy a variation on the strategy used by arbuscular mycorrhizae. They invade the lumen of plant cells and make specialized structures surrounded by the plasma membrane of the plants, and these fungi feed on living plant tissues. Bacteria that make nitrogen-fixing nodules on plant roots also penetrate into plant cells and form specialized structures called nodules that are surrounded by the plasma membrane of the plant cell. Nodules are involved in providing plants with nutrients, among other things. Little by little, after the emergence of the arbuscular mycorrhizal fungi, there was an increasing diversity of microorganisms that invade the lumen of the plant cells and form specialized structures. These structures surrounded by plasma membranes of the plant and that are involved in nutrient exchange with living plant cells. Here is a list of a few of them with their estimated dates of divergence. If you would like details about how the ages of these groups of microorganisms have been estimated, many are reported in a recent chapter in Annual Reviews of Phytopathology. For those that are not described in that chapter, I use the online tool at timetree.org to estimate them. At another level of intimacy between plants and microorganisms, the microorganisms stay outside of the lumen of the plant cell and reside in the space between the cells, the apoplast. The oldest known such interaction was achieved by the ectomycorrhizae. The first ectomycorrhizal associations occurred with conifers about 120 million years ago, and numerous independent divergences of other species that make ectomycorrhizal associations with plants have occurred since then. For tuber, Laccaria, and Amanita, it is believed that their ability to establish ectomycorrhizal associations arose after the ancestral lines of the fungi lost the genes for numerous carbohydrate active enzymes, or Cas. Cas tend to elicit host defenses. We might say that by losing these enzymes, the fungi no longer elicited the ire of the plant host, thereby making the plant more docile and friendly. Interestingly, in the case of Calotatricum tophiidae, Cas genes have not been lost from the genome, but instead their expression has been turned off. Most of the estimates of evolutionary age that I have presented up through the ectomycorrhizae are the results of numerous scientific studies as described in the Annual Review of Phytopathology chapter. This evolutionary timeline has been presented previously by other authors, so it's not a scoop. On the other hand, there is less information available on the time of divergence of plant pathogens. When I began compiling information from the few reports in the literature about divergence of pathogen species, with estimates I made using the timetree.org tool, I could see the flush of diversification that occurred after the emergence of the ectomycorrhizae. So it seems that over several hundred million years, endomycorrhizae had been really cozy with plants and had privileged access to the inside of plant cells. Only after that did the apoplast become the new microbial metropolis. Some of the groups of bacteria that colonized the plant apoplast diverged at about the same time as the ectomycorrhizae, but most have diverged much afterwards. and likewise for many of the fungi that colonize the apoplast. These are just a few of the many examples that can be estimated. For those of you wondering about plant viruses, very little is known about their evolutionary timeline. Retroviruses that are part of several families of long-terminal repeat retrotransposons are believed to have integrated into plant genomes about 120 million years ago. Diversification of the begomoviruses into two subclades occurred about 25 million years ago. But other than that, it is currently believed that the diversification of most of the existing viruses occurred very recently, probably not more than a few centuries ago. I could go on and on with a list of pathogens along a timeline. You can see a few more in the Annual Review of Phytopathology chapter, and you can calculate divergence times for others with the timetree.org tool. But with the short list I presented, you are probably already thinking about the question I raised earlier concerning the progression of interactions between plants and microorganisms. I will present one more example to illustrate a trend. The genus of bacteria called Streptomyces emerged more than 1,000 million years ago. Streptomyces were present on continental landmasses when the first plants arrived on land, and they probably played a role in helping plants adapt to land. I will talk about this in the next section of the presentation. There are a few species of Streptomyces that cause plant disease, most notably Streptomyces scabies, that causes potato scab. Streptomyces scabies is very recent and probably diverged as a species only about 150 years ago. One trait that typifies pathogenic species of Streptomyces is the production of the toxin thaxtamin that inhibits the biosynthesis of cellulose. This causes plants to generate abundant quantities of the intermediaries of cellulose synthesis that can be used as food by the bacterium. These examples suggest that the overall progression of interactions between plants and microorganisms has moved from the more intimate interactions, where one organism has fully entered inside the cells of the other organism, to a more peripheral interaction, where the microorganisms obtain resources outside of the plant cell. Although this progress has involved gain of function by microorganisms, such as acquisition of type 3 secretion systems or various toxins, loss of function by microorganisms has also facilitated these interactions. Overall, the striking aspect of this evolutionary history is that it was founded on openness and welcoming on the part of the plants, rather than on separation and so-called war. Perhaps this surprising direction of intimacy between plants and microorganisms was due to the nature of the cell surveillance systems that allowed plants and microorganisms to recognize and discriminate between self and others. Recent research on the evolutionary history of these systems illustrates that they have diversified significantly, and in particular since the advent of angiosperms, leading to major systems of defense in plants. Here I have listed a few of the recent publications on the evolutionary history of NLRs in plants and fungi. Through horizontal gene transfer, fusion, and truncation, the ancestors of these genes have given rise to the components of sensor and signal transduction systems used by cells in environmental surveillance, some of which are R genes involved in resistance of plants to various pathogenic microorganisms. The diversification of these systems might explain why the evolutionary trend of interactions between plants and microorganisms is toward more peripheral interactions. Nevertheless, the intimacy of the early interactions between plants and microorganisms has led to major advantages for all players. The interaction of land plants and microorganisms over the past 450 million years has led to some major outcomes for life on land as we know it today. For microorganisms, new habitats and new niches in and associated with plant tissues have emerged, giving microorganisms access to carbohydrates and other foods. For plants, interactions with fungi have led to the developmental processes that gave rise to roots and their architecture. Microorganisms have facilitated access to atmospheric nitrogen and to other recalcitrant minerals. Fungi in particular have played a role in solubilizing mineral rocks on continents. Some of the viruses that integrated into plant genomes have been responsible for expansion of genome size. Traces of this can be seen in modern plants in the abundance of transposons in their genomes, where about 75% of the maize genome and 25% of the rice genome consists of long terminal repeat retrotransposons, for example. These are the jumping genes for which Barbara McClintock won a Nobel Prize. Interaction of land plants and microorganisms also led to the creation of soil, mostly due to the activities of streptomyces. As I mentioned earlier, this group of bacteria emerged about 1,000 million years ago. These bacteria produced filamentous mycelia and highly resistant spores, a bit like fungi in terms of morphology. They also have tremendous metabolic capacities in that they can degrade chitin and cellulose, produce proteases, protease inhibitors, and more than 7,000 secondary metabolites. The likely role of streptomyces in the survival and adaptation of plants on land was to provide a bed or contact between rootless plants and rock. This allowed the stems of the plants to absorb nutrients released by streptomyces and for streptomyces to acquire nutrients or debris from the plants. In the introduction to this presentation, I said that my motivation was to try to frame differently our thinking about the interaction of plants and microorganisms in an effort to foster novel approaches to plant health management that is more rooted in ecology. So, how does this evolutionary perspective that I just presented give us insights into epidemiology, emergence, and management of plant disease? I will explore two questions as examples. The first question concerns the precautions that could be taken when expanding the range of a crop. I will use the example of eucalyptus. Eucalyptus is native to Australasia, but at present, the largest industrial eucalypt estate is in Brazil. The bacterium Erwinia psidi is causing an emerging disease problem on eucalyptus in Brazil. The strains involved in the disease are identical to those causing a disease on guava, which is an indigenous crop in Brazil. Could this host jump have been expected? Eucalyptus and guava are both members of the Myrtaceae family that regroups nearly 6,000 species of plants in tropical and warm temperate regions. Among these, eucalyptus has been subjected to a major expansion of cultivation regions that are way outside of its current natural range. Interestingly, although guava and eucalyptus do not currently have the same natural geographic range, remains of the earliest eucalyptus were found in fossils from the early Eocene, the geological epoch from about 56 to 44 million years ago, in Patagonia, at the southern tip of South America. This suggests that not only are guava and eucalyptus in the same family, but that at one time in their evolutionary history, they might have had overlapping geographical ranges. The fact that guava and eucalyptus are in the same family might not seem to be reason enough for a host jump. But there is reason to wonder if host jumps are particularly easy in the Myrtaceae family. This is because of the behavior of one of the major emerging pathogens of eucalyptus, the rust Ostropoccinia psidi. This rust was first reported on guava in Brazil in 1884. Its host range includes 500 species of plants in 73 genera in the Myrtaceae. It is now found on plants in South America, the US, China, New Caledonia, Japan, South America, and Singapore. In addition to Erwinia psidi, other bacteria have been reported to cause disease on eucalyptus elsewhere in South America and in Africa. These other bacterial species are both in the Pantoia genus, the most closely related to Erwinia of all the genera in the Enterobacteriaceae. What can we learn from this particular case that could be applied to the general question of limiting the risk of disease emergences as we expand the range of a crop? New lands are continuing to be transformed into cropping regions. Also, with climate change, the geographic ranges of certain crops will change, so the question of range expansion of crops is very pertinent to assuring food security. First of all, we might want to take a more in-depth look at the host range of pathogens and assess the breadth of host range to assure that it is indeed limited to the species and cultivars that are assumed to be the only hosts. To do this, the set of plants used could be chosen from among species of the same genus, in the same family and order, and then beyond, in an effort to determine if there is some phylogenetic relationship among the plants that are favorable to development of the pathogen. A phylogenetic signal of host range has been observed in published data about disease and pest occurrence, but I am not aware of studies to assess the phylogenetic signal using data from direct experimentation. In any case, this phylogenetic signal could indicate for a given crop which adjacent crops, weeds, wild and ornamental plants could reinforce survival and emergence of a pathogen and thereby provide information useful for designing and managing regional land use. Likewise, it would be informative to understand the similarity of host range for a pathogen species in the same genus or beyond the genus, as in this study of host range of the Sclerotiniaceae family. As in the previous case, this analysis was based on published reports of host range. Experimental tests of fitness and growth of these fungi in plants would add important complementary insight that could help anticipate jumps of pathogens from one plant species to another, and again be useful for designing and managing regional land use and for crop production practices to avoid disease. For the second case, I will suggest how we can identify geographic regions that have the greatest likelihood for new outbreaks of an emerging pathogen so that there can be early intervention to try to prevent disease. I will build this example around the bacterium Xylella fastidiosa. Xylella fastidiosa is transmitted by insect vectors and causes decline or withering of its hosts. It is subject to quarantine measures all over the world. The current major hosts that are under surveillance are grapevine, almond, oak, coffee, and olive trees. The main disease management strategy consists of surveillance of commercial trade and stopping movement of infected material, and surveillance in the field and then destruction of infected plants plus those in a buffer zone. In Italy, for example, the decline of olives affects century-old trees of great value and is too widespread for the recommended eradication to be accepted by farmers and by citizens in general who appreciate these trees. Could we use insight from the evolutionary history of Xylella fastidiosa to find other options for disease control? In particular, could we anticipate epidemics so that there could be early preemptive intervention rather than surveying for a situation where it is a bit too late to save the plant? The intriguing aspect of Xylella in studies of the evolutionary history of microorganisms is its age. In the work shown here, the branch that led to Xylella fastidiosa diverged from the branch leading to Xanthomonas about 700 million years ago. It could be argued that the calculation by these authors in 2004 did not include the full scope of genetic diversity of Xylella and related bacteria that is available today. But my point is not to insist that this estimate of age is precise. Rather, I want to illustrate how it could lead to new approaches to managing Xylella as a plant pathogen. Another intriguing characteristic of Xylella is its broad host range. It has been reported to be present in 560 plant species, in 260 genera of plants covering 80 families of monocots, dicots, and gymnosperms. In hundreds of these species, no symptoms have been reported at present. In these cases, Xylella fastidiosa lives as a so-called endophyte in the xylem vessels of the plant. So let's go back to our supposition that Xylella fastidiosa is ancient, maybe as ancient as arbuscular mycorrhizae. We also note that arbuscular mycorrhizal associations are very widespread, being present in all gymnosperms and more than 80% of angiosperms. This analogy could lead us to wonder if, due to the long period of possible opportunities to associate with plants, perhaps almost all vascular plants harbor Xylella fastidiosa, or at least are able to harbor this bacterium. How would this supposition be useful? It is useful because it can help orient the search for situations at risk for the emergence of decline due to Xylella. The search can focus on environmental contexts that are favorable rather than on positive detection of the bacterium. This is exactly what colleagues at INRAE in Avignon did. Based on field data of olive decline on the island of Corsica, they showed that the best predictor of regions at risk for emergence of olive decline was the water deficit. They then showed that all occurrences of Xylella in continental France were in regions with high water deficits. Therefore, as a strategy, we could identify the regions of the world where droughts are predicted to intensify. This is currently being estimated in efforts to mitigate the effects of climate change. And then we could identify the plants in those regions that are likely to be the most sensitive to water deficit. These could be the object of intensified search for xylella, with tools from both classical microbiology and molecular biology. Research on means to limit proliferation of xylella in plants and to boost vascular system function to resist water deficits could be focused on these plants in these contexts. I hope that this presentation has set you in the mood to ponder. Before ending this story, I would like to point out two other perspective questions on the horizon. Firstly, might we wonder how much of the life history of plant pathogenic microorganisms we are ignoring by focusing on pathogenesis and disease? We might be missing important aspects of their lives that would reveal reservoirs of inoculum or how they overcome host resistance. For example, Botrytis cinerea and other species of Botrytis are commonly found as endophytes in apparently healthy plants. This was a surprise to many pathologists. Likewise, the closely related species Sclerotinium sclerotiorum invades plants as an endophyte in tissues that are distal to where in the plant it is causing necrosis. Botrytis and Sclerotinia can alternate between endophytic and necrotrophic growth in plants depending on the balance of signals that favor autotrophy or apoptosis of plant tissues. These observations are part of a growing body of data that certain fungi can have multifaceted lifestyles, making it difficult to conclude that they are strictly necrotrophs or biotrophs or commensals, etc. These classifications are probably best viewed as phases of their lives across a continuum of possibilities, depending on the outcome of their genetic potential in a context of biotic and abiotic environmental factors. The second question is about the molecular dialogue leading to disease. We need to ask if it is really distinct from the molecular dialogue leading to commensalism or symbiosis. We can look to trichoderma for an answer to this question. Trichoderma is well known for its beneficial effects on plants, of improving plant growth and in particular root growth, all while protecting plants from certain pathogens. Trichoderma's interaction with plants are mediated by effectors. Like the effectors of plant pathogens, the effectors of trichoderma set off host defenses. This contributes to its efficiency in biocontrol of various diseases, both in the rhizosphere and on aerial parts of plants. The evolution of molecular dialogue between plants and microorganisms is often called an arms race. But this vocabulary is not inclusive. It orients the perception of plant-microbe interactions as a war, when in fact, over the long-term history of interactions between land plants and microorganisms for several 100 million years, there has really been cooperation. Would diplomacy be a more comprehensive and pertinent framework to understand the evolution of plant-microbe interactions? Not only would it be more encompassing, it could serve as a better framework for developing ways to manage plant health that are based on cooperation and symbiosis, and these will surely be trends in the future, rather than on eradication and elimination. This would be more compatible with modern efforts to reduce the use of pesticides in agriculture and to found plant health management on principles of ecology. Now I invite you to listen to a song that will provoke more thoughts about this subject. You say there's been coevolution. Well, hey, can you tell me when it all took place? Did it start in the cropping revolution, in a dance that they call the plant microbe arms race? So many are genes and virulence alleles, like the plants and the microbes at each other's heels. Is that really what it's all about? Is that really what it's all about? Is that really what it's all talk about big evolution from long ago.
So long ago,
since plants first came on the land,
they've been around for such a long time.
Receptor and signal convolution is a chatter of diplomats while they're shaking hands. Mutual benefits in nutrient exchange with a couple of cheaters who got into the game. Is that really what it's all about? Is that really what it's all about? Is that really what it's all about? All about, all about, all about, all about. The story seems so conclusive when some pathogens conquer cultivars.
It really caused a lot of lesions.
But habitats can be so elusive outside of the farm. What do we know so far?
What have we explored?
In rivers and glaciers and with algae so green, maybe there's pathogens that we've not yet seen. Is that really what it's all about? Is that really what it's all about? Is that really what it's